Well, welcome everybody. Thank you for coming. Len and I are are being invaded by the. Len and I are old personal and professional friends. We've been working together on basic problems in gene expression and developmental biology and disease biology for decades. And we've had the pleasure of having extraordinary trainees who, when we get them together, do remarkable things. And I think in the last 12 years, probably a dozen papers in prominent. And they've come up with a new discovery, one that we're really excited about, we want to tell you about today. And there are really two discoveries that have come together, and that's why we're doing this with Camp 4, company we founded. One is that we believe we figured out a role for regulatory RNA that is being produced from the regulatory regions of genes in the control of specific genes, an insight that really changes the paradigm of gene regulation, and I'll talk about that. And we believe Camp 4 has found a way to exploit the ability of ASOs, antisense oligos, to bind selectively to specific regulatory RNAs and tune gene expression up in the context of diseases where you have haploinsufficiency or low levels of gene activity. So I'll let Len start this by talking a little bit about the disease context.
Great. Well, thanks again, everybody, for coming. So I'm Len Zahn. I'm the director of the stem cell program at Boston Children's Hospital, and I take care of kids who have blood diseases or cancer. And I've been focusing most of my life on studying how blood cells are formed and also how blood differentiation occurs. And in particular, I'm interested in how cells have a certain fate. So for instance, you have a blood cell that can have a certain fate versus a muscle cell. They have the same DNA, but how are those genes actually turned on? And so one way that they're turned on is to have a master regulator, a DNA binding protein that binds to DNA and activates that specific program. And that DNA binding protein is cell-specific in terms of its expression. So for instance, when I was a postdoc, I cloned a gene called GATA1, and GATA1 drives the expression of every red blood cell gene and turns that gene on. So several years ago, we found that there are growth factors that are on the surface of cells, growth factor receptors that can get triggered, and this will lead to an upregulation of genes that are cell-specific. And this was very surprising. So for instance, if you take a growth factor and give it to a red blood cell, it'll turn on some red blood cell specific genes, whereas if you give it to a muscle cell, it'll turn on muscle specific programs. And so that was very surprising. And so I went to one of my best buddies, Rick Young, and we started to talk about how could this occur. And one of the ways that this could occur is that there are triggers of the cell surface that lead to signaling molecules, signaling DNA binding proteins. And we found that those DNA binding proteins actually landed in chromatin right next to where these master regulators are regulated. And they will allow the upregulation of genes. And this is really an interesting concept. And we wanted to think about, could this be used therapeutically? And so when Rick and I were talking about this, we started to think about forming Camp 4. And so the concept at the very beginning was to upregulate some of these genes that were made by specific tissues. And so as we started to do our experiments, we would start to make maps of tissues. And these are transcriptional maps of tissues. So for instance, we made one of the best transcriptional maps ever of the liver. And so we mapped where every DNA binding protein would bind to chromatin in the liver and how it would turn on genes, how these domains would actually fold together to be able to regulate gene expression. And we also looked at the RNA expression and even the primary transcripts of RNA and saw that there was a synergy of these activities. And there seemed to be regulatory RNAs that were actually made by these regions of the genome, these enhancers, and these types of things might be able to be targetable. And in thinking about it now in Camp 4, we came up with the idea that if you could have an oligonucleotide, you might be able to tune these regulatory RNAs and basically be able to upregulate genes or downregulate genes. And so that led us to testing the hypothesis. So why in the world would you ever want to do this, right? So in a number of diseases, you can have haploinsufficiency. So, what is haploinsufficiency? Well, in a genetic disorder, if you have a mutant allele, a mutant chromosome, and a wild type, a normal chromosome, you could have 50% of the output of that particular gene. And so it's possible that if you could get the normal copy of the gene to be upregulated, that you would actually fix these genetic diseases. And so that became very, very interesting hypothesis for us to test. And there are over 650 haploinsufficient diseases. This occurs in many different organs in the brain, the eye, the heart, the liver, muscle, kidney, and the immune system and the blood system. And this is a real problem for the doctors to be able to figure out how to actually take care of these patients. And it would be very simple if we had a technology to be able to develop. And what we were able to look at is that there's a lot of different diseases in these organs that could be fixed by this particular type of technology. And you can see particularly the liver and the brain here. And the reason that we pick those in general is that the oligonucleotide platform by many other companies has been able to be perfected to use oligonucleotides to block gene expression. Now we want to use oligonucleotides to upregulate gene expression. But this could also be used for many other different disease states, such as the heart, the muscle, and also in the eye and other tissues, because any of these principles would be able to be seen. So I think what's amazing at Camp 4 is that through the technology that Rick will actually be able to discuss, you end up you end up being able to find a regulatory RNA like an enhancer RNA. You could target it with an antisense oligonucleotide and upregulate gene expression, and you can upregulate gene expression to a point where it would be therapeutically useful. And we've been able to show not only that you can just upregulate RNA, but you can upregulate the protein levels in multiple different species, including in primates. And then you can also be able to fix a disease model and have a physiological response. So, for instance, there might be a metabolic phenotype that's actually corrected by these oligonucleotides. We've been able to show a physiological response from some of our oligonucleotides. And I think that's quite remarkable. So to me, this is almost a paradigm shift for how you could treat these types of diseases. You're able to upregulate gene expression, and there are specific rules on how to do that. The oligos can be constructed in certain ways. We have AI to help us be able to design those oligos. And this is a way of approaching these that we haven't been able to approach it in other ways. And I just think that it's so cool to have a company that was able to make this discovery and be able to actually be able to upregulate genes. Now, the other thing I would say is that this is a gene BDNF, and that's a gene that's a growth factor. And because you can upregulate genes, you could also be able to upregulate growth factors. And that might also be something that would be useful therapeutically for a number of different human diseases. So I think Rick and I are very excited about it. It's an exciting time for the company, and we thought that it would be really great to explain the technology. And so I will turn it over to Rick.
The one thing I'll add to Len's comments is that, in principle, you can fix haploinsufficiency with gene therapy. But one of the things that we're very concerned about is that there are many diseases where overexpression of the product is just as deleterious as underexpression. So what you'd like is a natural system taking advantage of the way the biology actually works to upregulate genes to just satisfy this haploinsufficiency phenomenon. So to do this, let me explain a little bit about how we think about the way that gene regulation works. And I just want to introduce the fact that this work, which will be published in a little over a week, has a background to it. And the key folks here who've done the science are John Henninger, Oscar Oxus in my lab, Bob Warnford Thompson in Roberto Bonazio's lab, and Ann Robertson in Len's lab. So the picture you're looking at is the view we have of DNA coming out of the nucleus, and there are two key regulatory elements. They're the enhancers and the promoters. Most genes that are expressed in a cell type specific fashion have enhancers that are cell type specific. They're DNA elements that are bound by transcription factors, cofactors, chromatin regulators, and the like. And you have promoters. The promoters are the element that represents the beginning of a gene. They can also be bound by apparatus, key to the regulation of the gene. So the textbook model's here. The textbook model is there's a collection of transcription factors that bind an enhancer. Typical enhancer might encompass 200, 300 base pairs and be bound by 40 or 50 DNA binding transcription factors. Cofactors, chromatin regulators come in. Then they're going to recruit the rest of the transcription apparatus. And the paradigm is that the biggest class of genes are the genes that encode these transcriptional regulators. They have a DNA binding domain and they have an activation domain. They're separate domains, have these two different functions. However, what we've known since Phil Sharp, who's in the audience here, published it, is that RNA is being transcribed from regulatory regions. So it's being transcribed from both the promoters and the enhancers in a way that doesn't produce a full-length pre-mRNA, but rather is turned over very rapidly. But we haven't known what it does. There have been many hints that it has function, but we didn't know what it does. And the new paradigm I'm going to introduce to you is that the majority of these gene regulators have a third binding domain that binds RNA. And what is happening at these enhancers and promoters is you are getting large assemblies. You are not getting one molecule of RNA polymerase, one molecule of a transcription factor, one molecule of a coactivator. You're getting very large assemblies that form what are called biomolecular condensates. And condensates are regulated by RNA. And indeed, what we think is happening is that the enhancer RNAs, and perhaps RNAs adjacent to the promoter, are contributing to the regulation of each gene. They're doing that because they'll regulate the behaviors of these biomolecular condensates. They're doing it because the transcription factors fall into two classes. They're activators and they're actually repressors. So there are regulators that do both. These RNA molecules can bind regulators selectively. In other words, although there's promiscuous RNA binding by these regulators, there's a preference for a specific sequence by each positive factor and negative factor. And we've seen strong evidence that what the RNA is doing in addition is it's helping retain the regulators in this chromatin space where gene regulation is occurring. What this leads to then is an ability that CAMP4 has now demonstrated by a very large number, dozens of disease genes, that is essentially in every case, they can identify an ASO targeting selectively a regulatory RNA, an RNA from the enhancer or from the promoter, that we believe has the effect of selectively removing the repressor. And we believe that's the mechanism by which they're able to demonstrate over and over again this ability at essentially every gene to upregulate the transcription of the gene, and as Len pointed out, to actually in model systems have a physiological effect that would be beneficial in a patient context. So I'm going to turn this over to David Bumkrot, who's just going to add a few comments.
Thanks, Rick. Thanks, Len. So I am David Bumkrot. I'm the CSO. So you just heard about the science. RNAs. Thank you, Felicia. As I was saying, I don't know if it's on or not. I'll assume it's on. Um, so Camp4's platform does two things. One is it identifies the regulatory RNAs that are associated with whatever gene we want to upregulate. And then the second thing is we identify the antisense oligonucleotides, which we then optimize to increase expression of the disease-associated gene. And as Len explained, the pipeline that we're currently building focuses on genes in the liver and also in the CNS areas where ASOs are most successfully being delivered. And our first program in metabolic disease should enter the clinic the end of this year or early next year. But as we're doing that, and to fully, I think, take advantage of this science, we're actively seeking partnerships to exploit this in other tissues. With partners who would bring expertise in delivery and in disease biology and other tissues. So with that, I would like to open the floor to questions to our speakers, and thank you all for coming and watching online.
I should defer to some of the wonderful scientists here who probably will ask much more intelligent questions than I will. But I wanted to get a sense for what these oligo structures might look like. Do they have you mentioned some rules that you were starting to learn? You know, is there a certain length? Are there modifications that you need to be made like other oligonucleotide therapies that make them more drug-like? Are there either encapsulation approaches or conjugation, pardon me, that enhances delivery, especially to the tissues that you've mentioned? Just a couple questions along the characteristics of the ASO therapeutics themselves. Thank you.
So I think one of the real advantages we have is that the oligonucleotides that we're developing so far really have the properties of ASOs that have already been in humans, that are either approved drugs or are drugs in late-stage clinical development. So for example, for the liver delivery, we take advantage of a carbohydrate ligand, GalNAc, that Alnylam, of course, uses, as do other companies. It gives very efficient uptake there. In the CNS, intrathecal administration of a stabilized oligo is sufficient to get remarkably broad distribution in the CNS. The chemical modifications are the ones that are found in known ASOs and that are available to us and that can be manufactured at scale. To get into other tissues, I imagine we will explore some of the things that you talked about, whether it's encapsulation or other ligands going forward.
So I'm really excited about the insight that targeting these RNAs will affect regulation. But I am surprised that it is relieving a negative regulation or facilitating a positive response, as contrasted to being either negative or positive, depending on the context. So is it clear that is there biochemical changes in the promoter structure, chromatin structure region that correlates with the effect of stimulation of transcription with these oligos? I mean, the biology is a little strange.
David, you should answer that. Your team has the result with the evidence that they've.
Yeah, I think one of the things that was surprising to us is that it turns out that when we make a series of oligos across a regulatory RNA, depending on where it binds, we can sometimes get either an increase in gene expression or a decrease in gene expression. So far, we don't really have any compelling evidence, regardless of the structure of the oligo, that we're actually targeting the RegRNA for degradation. We believe these are acting in a steric manner, and we have some evidence, at least for one gene we're working on, for a decrease in binding of negative acting factors measured by ChIP-seq or ChIP-PCR, HDAC and NCOR1 family members, that's consistent with the mechanism that Rick just described. But definitely it needs to be more fleshed out.
So thank you. I'll follow with another question. You've talked about enhancer associated and promoter associated RNAs. Do you see the same phenomena with long non coding RNAs in the vicinity of a gene?
Yeah, so these other RNAs, some people call them natural antisense transcripts or NATs. That's been a term we've looked at somewhat. We're focusing more, I think, on the types of RNAs that Rick talked about. We have at least one example where indeed we were able to upregulate a gene by targeting one of these long non-coding RNAs. You know, and I don't know if it's the exact same mechanism, to be honest.
Yeah, so in my world of gene therapy, it's the delivery stupid. That's always the point. And I'm just curious. It seems to me that the scientific principle here makes perfect sense.
Mhm.
But the question is whether oligonucleotides are going to prove to be generally useful enough in terms of percent of the cells that you need to hit. We see that CNS diseases with AAV, you see it all over the place. And I'm curious whether or not there's any interest, or I know how difficult it is to try to develop small molecules that will do the same thing, but it kind of seems like everyone wishes that there was a small molecule solution to this, because if this doesn't work, it'll probably not work because of the concept. It'll work because you're not delivering it to a sufficient proportion of those cells. Liver may be an exception. CNS, I don't think will be.
I think that's an interesting point, Richard. I think at a minimum, these ASOs, which can be drugs, provide us with an assay that one could then use to develop a small molecule that might have the same effect. And as you know, there have been cases where ASO therapeutics have been replaced by small molecules that have the same effect. So I think that's an interesting point. That's a longer term issue to get to the clinic, but that would be an interesting possibility.
I still think, though, you are in the few examples we've looked at where you do get a physiologic response. I don't think it could be explained by a small number of cells responding. And some of those are liver, so I think that that's fair. But I do think that some of the responses we see have to involve a lot of the cells that are affected.
I think a lot of the diseases you're going after are monogenic haploinsufficiencies, right? But the mechanism is ASOs binding RegRNAs to unleash transcription factors, which don't just drive one gene all of the time. So are there limitations to what RegRNAs you can target or what haploinsufficiencies you can go after because of the ubiquitous nature of some of these transcription factors?
From my perspective, I mean, well, David answers part of this, but you know, I think we've been able to examine adjacent genes and see what's going on with other. It seems relatively specific to the gene you're targeting so far, and I think we've targeted over 30 genes and been able to upregulate over 90% of them. So I think that the data looks very good at the current level, but I don't know if you have a specific, anything more specific than that.
Yes, that's right. So it really looks like these RNAs act locally in three-dimensional space near where they're being made.
And I think that's one of the advantages is you have a specific oligonucleotide, you're really affecting just that gene, and so you're not affecting other genes. And you know, that's why small molecules, which was one of Camp4's idea at the very beginning, you know, affected more genes. These seem to be pretty specific so far.
Just building off of the previous question, we can design it to be fairly specific and target one gene of interest. But I wonder, based on Rick's comments, could it be possible to design these oligos to alter the properties of some biomolecular condensate? And then you can start altering the expression of a network of genes, and could that have some value?
Well, Anchor, you're the expert in that. So I guess you're suggesting that because you believe it's a possibility. And one of the features I find exciting that I didn't talk about is, of course, now that you know that transcription factors have a DNA binding domain, an RNA binding domain, a protein binding domain, It can be that they're brought to these sites through their interaction with RNA as much as with DNA. It can be that some of the GWAS variants that we see having the propensity to develop disease are actually acting through a change in the RNA molecule as opposed to the DNA molecules are binding.
Sounds like there's a question coming in from our online viewers. The question was about applicability beyond genetic haploinsufficient diseases. And yes, there are a great many. We think of this as sort of, where gene augmentation would be applicable. So increasing expression of a gene, even if neither of the alleles is mutated. And indeed, that's the approach that we're taking in one of our first programs in metabolic disease, increasing expression of a component of a biochemical pathway that's sufficient to overcome partial loss of function mutations even downstream of that gene. And so that's another major class of diseases that we're working on. Good question.
I think the example of the growth factors would be another one. Is certainly if you needed to express a particular growth factor, that would be helpful for a number of immunologic diseases, blood diseases, that type of thing would also be for many different types of diseases. So I think it would work.
Question from the back row. So we started off the conversation talking about these regulatory centers as being really important in driving cell identity and categories of cell identity, but yet you've been able to see activity with over 90% of genes that you've tested. So are there subcategories of genes that are more amenable or less amenable? And does that correlate well with the genes you'd like to target in disease?
I'll let David do that.
So there likely will be categories of genes that may be less amenable to our ASO approach. I don't think we know enough yet to beforehand be able to predict that. But what's developing is that there are some genes that maybe fall within the class of what you might call housekeeping genes, or genes that are expressed at a very high constant level, not subject to regulation, if you will, that may be more difficult for us to modulate. But this will be a minority of disease-associated genes.
More.
I'll get one more in.
Awesome.
Might be a very unfair question and probably one from the analyst community, but compared to something like an ASO for exon skipping, which has a set level of efficiency, which is pretty poor, to be honest, how does this system sort of compare to that? If I noticed one of them was for DMD, so we pick exon 51. How would you sort of compare to exon 51 or something like that?
Yeah, I don't think we have the experience yet to compare them directly to each other. So, but I would say what we're seeing is a robust upregulation of the genes. Exon skipping is a valuable set of drugs to take, and even though some of them work and some of them don't work, there are still many companies that have them. So I don't want to bash that, but I do think that this approach is relatively new, needs to be compared to the other approaches and see what happens.
Well, thank you very much, everyone, for attending. We really appreciate you being here.
Thank you. Cool. Thank you, David.